thank you for for introduction. So good afternoon, everyone. My name is Lin. I'm a rising junior at Virginia Tech and majoring in nanomedicine. I work with computational microscopy image laboratory at the University of Florida with Dr. Pinaki Sadar during the summer. Today, I'm going to introduce my project, Interactive Spatial Transcriptomic Visualization Portal, to you guys. So first, let's discuss the most important thing of this project was spatial transcriptomic. This will use a tool called Visian Spatial Gene Expression. This tool will measure the total RNA in the whole tissue section. On the Visian Spatial Gene Expression slide, there are capture areas with spots containing the special RNA capture molecule called primers. And the primers will capture the RNA at those spots. And the captured RNA is converted into cDNA, which acts as a copy of the RNA. And each cDNA for each different spot will receive a unique special barcode that just like a special ID so that we can easily use it to do some search. Next, we will create the library and read them to determine the active gene in each different spots. We will use the barcode to connect the gene activity data back to the precise location in the tissue section. That will enable us to create a map that will showing where each gene is activated in the tissue. So in the spatial transcriptomic, the spot can have coverage of multiple cells, and the cell within the same spot can have different cell type. In order to find out the cell type they are, we can do the enrichment analyze to find it, and we also can do the deconvolution to obtain the cell type proportion. So what is a cell deconvolution? It's a computational pipeline that can be used on spatial transcriptomic data to predict cell type proportion. You can do this because DNA can be transcribed to RNA and then transcribed to and translate to become amino acid. And then it will fold and become protein, and you can get in the signal from other cells, work with other organelle, and you will finally become the tissue and become the organ that we can see. And that's the phenotype we have. And the gene inside everything will be the genotype. And because of this whole process, we can use the gene expression pattern or the genotype to predict the cell identity or the cell type or the phenotype at every different spot. So in summary, we will have all the essential input from the previous step. They will store with different components with the Sarat object, and those components will form the foundation for our spatial transcriptomic data structure and play a crucial role in the cell deconvolution. So the first step we will do is to obtain the raw gene expression data, which will represent the gene expression level for each gene in every cells or spot within this tissue section. And that will be stored with the assay components. And after we obtaining those data, the next step will be getting each cells or spots with its corresponding cell type and cell state information, which will be stored with the metadata. And after we have those two data available, we will apply a dimensionality reduction technique to transform the high dimensional gene expression data to a low dimensional space. And we often use PC and UMAP to do this. And those results will store with the reduction components. And finally, we will have the image information such as spot coordinates and scale factor. And both of them will integrate into the analyze, and the image information is essential for overlaying the gene expression data on the tissue image and gaining a spatial perspective of gene activity within the tissue section. And that will be stored with the image components. So the objective or the purpose of this project is to make spatial transcriptomic data accessible to all. It will make people quickly getting the predict cell type with different tissue section and give user powerful visualization and interactive tool. This portal will have intuitive and user-friendly interface that will ensure that every user with different background can easily access and export the visualization. And people can also use it to analyze and interpret the spatial transcriptomic data efficiently. And one of the highlights of our portal is the seamless visualization of cell deconvolution image for select cell type. This feature will allow users to understand the relative predicted expression level of different cell types within the same sample. So there are a couple of methods that we use to create this kind of portal. So Shine is the first package that we use. So it's a web application framework that allows us to build the interactive and user-friendly GUIs directly from R. It provides an efficient way to create the web-based interface for data visualization, analysis, and exploration without the need for extensive knowledge of web developed language like HTML, CSS, or JavaScript. And there are two main components with Shine. So the first one is called user interface. It will use to define the layout and appearance of the web application that user interact with. In this case, the UI compresses various elements such as the titles, the plot output, and file upload dialog, the interactive control like drop-down menus and buttons. And second component is called server. It has the responsible for all background job or data processing, logic, and handling of user interaction within the web application. It will take the input from UI and perform the computation or operation using R. For example, we can read and pre-processing the spatial transcriptomic data, perform the cell deconvolution, and generating the interactive plots and visualization. So the second method or the second package that we use is called Sarat. So Sarat aims to enable users to identify and interpret source of heterogeneity from single-cell transcriptomic measurement and to integrate the diverse type of single-cell data. Here are two important functions that I have learned. So the first one is called C-transform. This is a pre-processing method that can use to improve the quality of single-cell RNA sequencing data with three components. So the first one is called normalization. We will apply the size factor normalization to the row count data. This step will ensure that the data is on a common scale. And then we have batch correction. We will use this to integrate the data from different experimental batches. And lastly, we have virus stabilization. So after batch correction, we will use this to ensure that the variance of gene expression is constant across different expression level. And the other function that I have used is called spatial feature plots. So this function will provide a spatial views of gene expression pattern across the tissue section. It will allow us to select a specific gene or feature of interest and visualize their expression pattern at different spatial locations within the tissue. And you will see a similar example like this on my portal. So this is just basically showing the expression level for the specific gene expression with different colors. The blue will represent the lowness expression level and red will represent the highness. And in this portal, we will use uniform manifold approximation, which is called UMAP, to visualize the high-dimensional transcriptomic data. So the UMAP is a dimensionality reduction algorithm or tool that enable us to reduce the complexity of the data while preserving its underlying structure and transform the data into two dimensions right here. So in order to generate UMAP plots for a specific cell type like this, we will select the cell type of interest first, and the portal will filter out the data to include only that cell type, which will allow us to focus on its unique expression pattern within that tissue. And each point on the UMAP plot will represent a specific spot within the tissue section. And one of the key features of our UMAP plot is it has the ability to obtain the expression level of select cell type. The color of each point on the plot will correspond to the expression level of the chosen cell type within the spots. The color coding will help us to quickly identify the region of high or low expression for the select cell type. So based on everything that I just said previously, I have created a video for how to run this portal. So I have run this code and you will run the whole background program. And I have uploaded a RDS file which contains the Sarat object and start visualization. And as we can see on the left side, it's the UMAP plot that we just mentioned. And the color will correspond to the color on the right side, which means the predict expression level for a specific cell type around the tissue. And here I have chosen a specific cell type as an example. You will see there's only one red dot on the right image. This means this spot has a very high expression level. And it's also easy for us to find out the specific points from the UMAP plot since there's only one red dot. So I will click on that spot and then you will see an arrow points to where the spot is with the cell deconvolution image. And it will also give you the coordinate information for where it is on the image for that specific spot and the barcode information below. And lastly, I have created a place to upload a TIFF image, which can be the histology image so that we can use the histology image to compare with the cell deconvolution image in order to find out what happened with the phenotype, which we can see from the histology image. Compare this with the expression level on the cell deconvolution image. So based on what I have for the results, here's the discussion for the expansion for the current prototype. So firstly, I will say, as you can see from the result parts, I can be able to upload the histology image. We want to add it to allow user to zoom in and zoom out in order to get more detail with the high resolution so that we can see more detail with the single spot, just like what this map gives showing. And we also wanted to zoom automatically to where the select spot is when we click on the UMAP plots. And the other thing that we can do is, because we can get the coordinate for where the predict cell type can be, we can easily find out the place for the cancer cells so that we can set it as a target for the drug to attach. So lastly, I would like to say thank you to Dr. Pinaki Sadar, Salman Brother, Myers 10, Ahmed Nagla, Ellen Danpour, and all other members of the lab for helping me to learn different things and figure out the problem that I have for my project. Also, I'd like to say thank you to Dr. Zarina, Dr. AJ, and Maya for their support. And thank you to the HuBMAP program and Internet Edge for funding and for giving me this opportunity to learn more. Thank you so much for your time, and any questions?